we became very interested in measuring viscosity on a single-cell level. Essentially, viscosity determines how fast species can diffuse together, so in a cell that can affect many important processes such as signaling or reactions. Also, there's some evidence that abnormal level of viscosity can be linked to disease or malfunction. So determining viscosity, especially in a single cell, could be very, very interesting. For us, the main challenge was to figure out how to measure the viscosity on a single cell level, because on such a small scale, conventional mechanical methods are not going to work. So our approach to deal with this was to use the small fluorescent molecules called molecular rotors, in which fluorescence, emission of light, competes with intermolecular rotation when one part of the molecule rotates versus the other. And in a viscous environment, this rotation is going to be slowed down, so the competing channel, fluorescence, can be very dramatically affected. So by measuring either the fluorescence spectra or the fluorescence lifetime of molecular rotor, we can very well determine the viscosity. We have discovered two such molecular rotors, and both of them show very large changes of fluorescence with viscosity. And both of them are biocompatible, so we can put them inside cells and then measure fluorescence coming back from cells and create maps or images of intracellular viscosity. We need to remember, of course, that maps of viscosity in a cell can change with time. For example, if cells become stressed or diseased. And our method based on molecular rotors allows us to detect such changes, even if they're quite fast. So we tried to measure how viscosity changes in a dying cell during light-activated cancer treatment called photodynamic therapy. Photodynamic therapy works by killing malignant cells upon irradiation of the drug. Without irradiation, drug is non-toxic to cells, but when irradiated, it starts producing very toxic, very short-lived intermediates, molecules inside the cell, which then lead to cell destruction. Using our molecular rotor, we managed to discover that in a cell subjected to photodynamic therapy, viscosity increases dramatically as the cell dies. And essentially that means that in a dying cell, some reactions which require two things to come together and react, and perhaps some signaling events, are much, much slower than in a normal, healthy cell. So we hope that this method, which we developed and used, will help us learn much more about the intracellular environment, and also perhaps understand how some diseases and their treatment can affect biological function.